Thus far, we've discussed the structure of DNA, the structure of chromosomes. We discussed DNA replication and how that relates to the cell cycle as well as mitosis. And now we're going to talk about loss of cell cycle control and how that leads to mutations and ultimately to cancer. For the average human, they will have some 100 trillion cells die in the process of their lives. We change all of our skin once a month. So that is a lot of proliferation and a lot of cell death that has to take place just within our skin. We are shedding something along the lines of 30,000 skin cells per minute. So with regards to our skin, we are constantly regenerating cells. We can think of cells having one of three primary fates. It can either go into cell division, which is the process we've been talking about, where you undergo DNA replication, and that leads to mitosis. So we end up with two daughter cells when we started with one. You can also, as shown in B, enter a process called differentiation, and that's where a stem cell enters into a process where it can become a well-defined other cell type. Lastly, a cell sometimes needs to undergo cell death, and that's shown here, showing apoptosis. For this portion of our talk, we're going to mainly be focused on this process of cell division, again, which is mediated by the process of mitosis, which we talked about in the preceding lecture. What we are doing here is starting with an original parental cell, and the end result of mitosis is that we have two daughter cells that appear identical to the parental cell. Now, once a cell undergoes division, it's going to enter the cell cycle, as we've discussed, and this cell cycle is very tightly controlled. We have a series of checkpoints in the cell cycle to make sure that as the cell enters this process, cell division is undergoing properly, that we have the necessary nutrients to undergo the cell cycle, and that the cell has properly divided. We have three main checkpoints in G1, G2, as well as in mitosis. Those are shown here. Our first checkpoint is going from G1 to S. The second checkpoint is in G2 as it's transitioning prior to going into mitosis. There's also a key cell cycle control step in mitosis at the metaphase plate. In G1, what the cell is needing to assess is whether or not you even need to undergo cell division. This process requires lots of nutrients to take place, so the cell is asking itself, are the necessary growth factors there to undergo this rigorous process? Is the cell even big enough to withstand doubling of all the DNA as well as doubling of all the organelles? Once that decision has been made to proceed into the synthesis phase, it will then move on ultimately after all the DNA has been replicated into G2, which is another growth phase. And in G2, what happens is there's an assessment of whether or not DNA replication, which happened in the S phase, actually occurred appropriately. Again, the cell needs to grow, and there's an assessment of whether or not the cell is large enough to undergo the process of mitosis. Once it's entered into mitosis, at the metaphase plate is another serious checkpoint in which all the chromosomes have to be attached to the microtubules appropriately, otherwise you'll get an inappropriate number of chromosomes in one cell versus another cell, such that the end result will not be two identical daughter cells, but daughter cells with different numbers of chromosomes related to one another and different from the parental cell. So again, the checkpoints within the cell cycle, we have that first checkpoint at G1 to S. The cells will arrest here, they will stop here, unless the cell has enough nutrients or it's big enough. If indeed things are not going well and there's not enough nutrients, eventually the cell can enter into the process of programmed cell death. At the G2 to M, again, this is the second major checkpoint. They're assessing whether or not DNA was replicated appropriately. Is a cell big enough, and is there a favorable environment? If there's been major errors in DNA replication, the cell, again, can enter into a process of cell death. Lastly, in mitosis, at that metaphase plate, the cells are not going to move through mitosis unless the chromosomes are fully condensed and whether the cells are lined up properly and attached to mitotic spindles. If they are not, again, the cell can undergo cell death. So as we've now covered, there are three key cell cycle checkpoints at the G1 to S transition, G2 to M, and lastly in mitosis. Now, what does all of this, the cell cycle, have to do with cancer? Well, what we know is that loss of control of the cell cycle ultimately does lead to cancer. Most cancers are due to mutations in various proteins and enzymes that are regulating that G1 to S transition. So at that particular checkpoint, there's very important analysis being done of whether or not the cell should actually proceed into the synthesis phase. And if you have mutations in the genes regulating that checkpoint, what you have are cells going into the cell cycle inappropriately and leading to rampant cell division, even though those cells should not be dividing. Really, what we can think of cancer is just a problem of cell growth. We are over-proliferating our cells and not killing as many cells as we should. So cells that shouldn't be proliferating are proliferating, and cells that should be undergoing cell death are not dying. The end result is a massive accumulation of cells. What are the different types of genes that control cancer progression? There's two main classes of genes that you must keep in mind when we talk about cancer. The first are proto-oncogenes. So proto means before, and oncogenic indicates cancer. So if you think about an oncologist, that's a cancer doctor. So this is before the gene is actually cancerous. So for a normal non-mutated gene, it has the potential to become oncogenic. The proto-oncogene usually plays a role in cell division or differentiation. And oftentimes we find proto-oncogenes regulating that G1 to S checkpoint. So normally it's regulating what genes are allowed to enter into synthesis, but for a mutated gene where the proto-oncogene becomes oncogenic, it's now no longer regulating that checkpoint, and cells are going into the cell cycle when they shouldn't be. The second major class of genes involved in the progression of cancer are tumor suppressors. So these are the genes that normally are able to cause cell death, apoptosis, the programmed cell death. So normally in fully functioning cells, these genes are able to recognize mutations in the cell or when DNA has not been replicated appropriately, and they will cause cell death. They'll recognize that the cell is not healthy and should be aborted. However, if you mutate a tumor suppressor, the suppressor is no longer able to suppress the tumor, and hence you get tumor progression. So let's look at these two types of genes in a little more detail. Again, as I said, proto-oncogenes are normally genes that code for the cell cycle control proteins. When these genes mutate, they become oncogenes. So once they've mutated, they're no longer precancerous, they are cancer-causing genes. They no longer properly regulate cell division, which means that cell division is overstimulated. Cells are entering into that cell cycle and undergoing cell division more than they should. So if you were to go back to that initial diagram I had, what you see is that that single cell is over proliferating and you're generating a huge, massive accumulation of cells when you shouldn't be. Whereas we know that proliferation is due to a mutation in a proto-oncogene, we also recognize that a mutation in a tumor suppressor can also lead to an accumulation of cells. So cells that should be undergoing apoptosis or programmed cell death are not dying. Again, the end result is this huge, massive accumulation of cell growth. So these tumor suppressor genes are normally proteins that stop cell division if the conditions are not favorable. When mutated, however, cells are able to override these checkpoints. So cells that should be entering into cell death because they were not replicated appropriately, for instance, are now replicated and multiplied over and over again, such that we end up with a massive accumulation of cells. So in summary, for this portion of the lecture, what we need to recognize is that proto-oncogenes, when mutated, become oncogenes, and the end result is cell growth. Tumor suppressors, when mutated, causes inactivation of the tumor suppressor such that it's no longer able to suppress the tumor and no longer able to cause cell death. The end result is also cell growth. So in order to get the cancer phenotype, we have to have a mutation in both proto-oncogenes as well as suppressor genes. So let's say that that has been done. What does cancer actually, what is the cancer phenotype look like? Well, initially it's going to just be uncontrolled growth with a mass of cells. However, if they stay in one location, we consider that a benign growth that can usually be excised without little damage to the surrounding tissues. However, over time, as more and more mutations accumulate, that cell will begin to overgrow the surrounding tissues and begin to invade its neighbors. At that point, it's considered malignant. It becomes a little bit more challenging to actually remove that, and it's now considered a cancer. Once cells begin to migrate and invade other tissues, it's considered metastatic. So to get cancer, you have to have mutations, and many mutations accumulate in one cell, and this happens over a long period of time. So as you see, as we age, the likelihood of many mutations occurring in a cell type goes up dramatically, and this just takes time. So this is just showing it from a figure in your book. We have a normal cell undergoing normal cell division. However, a single cell can begin to accumulate certain mutations such that this leads to aberrant proliferation. At this point, it's localized and can potentially be removed. However, if the cell continues to undergo additional mutations, that can lead to the malignant state, at which point it's considered cancerous. Once a cell detaches from that initial tumor site and begins to migrate through the blood or the lymph to other parts of the body, it's considered metastatic, and this is much harder to treat. Again, I want to reiterate that to form this cancer phenotype, to develop cancer, requires that you have many mutations. We call this the multiple hit model. So the process of cancer development requires several mutations within a particular cell. We often say somewhere around 6 to 7. Some of these mutations can be inherited from birth, so you might have a familial risk for a particular type of cancer, but it doesn't necessarily mean that you will get that cancer. Most of a person's mutations are acquired during a person's lifetime, and this is the environmental impact. As I said, you usually need 6 to 7 mutations and key mutations within a cell for cancer to be induced. And this process also must include mutations of both a proto-oncogene as well as a tumor suppressor. So once you have the cancerous phenotype, how do cancer cells actually behave? Well, one of the first things they do is tumor cells will begin to generate their own blood supply. This is called angiogenesis. So here you can see a blood vessel, and the tumor site will release a signaling protein called VEGF, or vascular endothelial growth factor. Once that message is received by the blood vessels, they will realize that they need to make additional supply of blood to the tumor, such that the tumor can now feed itself based on this newly generated blood supply. This causes massive growth of the cells. A second property of cancer cells is that they lose their ability for contact inhibition. So normally, if you look at the top set of cells, cells will proliferate to a certain step, certain density, and then they'll stop growing. However, for tumor cells, they will proliferate, they will fill a dish, and then they actually start piling on top of one another. They don't get that signal that they're supposed to stop growing and that the space is full. Additionally, we say that cancer cells become anchorage independent, or they have a loss of anchorage dependence. So now what we know is for a normal cell, cells will only grow when attached to a particular surface. So for the cells on the left, the normal cells, they will only grow on the bottom of the flask. However, for cancerous cells, they're able to grow in the media as well as on the bottom. So they do not require that the cells be attached to a particular surface. And this is what we see with metastatic cancers, in which cells are able to travel through the blood and the lymph to other locations within the body. The last thing we know is that cells become immortalized. Cells no longer have a fixed number of cell divisions, and this is due to altered telomerase activity. Our telomeres are the ends of our chromosomes, and over time, as cells replicate, our telomeres get shorter and shorter. That is, unless we have telomerase activity, which is something that stem cells usually have, is high telomerase activity, in which the telomeres are extended repeatedly. As we age, our telomerase activity goes down. However, for immortalized or cancerous cells, they find that the telomerase activity is actually elevated. So one cancer target that is being pursued right now is various companies are looking at targeting that telomerase activity to inhibit cell division that way. So in conclusion, for our discussion of cell cycle control and mutation, we talked about the multi-hit model of cancer, such that you need 6 to 7 mutations in a particular cell for it to become cancerous, and 2 of those mutations have to be in a tumor suppressor gene as well as in a proto-oncogene. And lastly, we talked about some of the properties of cancer cells that make them challenging to treat. That includes angiogenesis, in which they generate their own blood supply, the loss of contact inhibition, the fact that they become anchorage independent, as well as having increased telomerase activity. Thank you so much.